everyone. In today's video, we're starting a new section. We're going to cover chapters 19 and 20. So chapter 19 is all about proteins, and then that will lead us into chapter 20, which is all about enzymes, which are a type of protein. So before we can talk about proteins, we have to talk about what makes up a protein. And the building blocks of a protein are amino acids. So amino acids are shown, well, the basic structure is shown down below. So notice that we have this carboxylic acid group. Although in this case it's missing its hydrogen because we're assuming that we're in a slightly acidic solution. So the carboxylic acid kind of gave up its proton there. And then we have what's called an alpha carbon in the middle. So the reason we call this an alpha carbon is if you have a carboxylic acid, so let's just draw one here. And let's say there's a chain of carbons coming off of it. We tend to label the carbons in the chain alpha, beta, gamma, etc. So the first carbon next to the carboxylic acid is alpha, the one after that is beta, gamma, and then we just keep going down the Greek alphabet. So here we call that carbon next to the carboxylic acid the alpha carbon. All right, and then next to that we have the amino group, and this is where amino acids get their name because of that amino group. We also call amino acids alpha amino acids because the amino group is attached to that alpha carbon. Now, the alpha carbon is also bonded to a hydrogen, and then finally an R side chain. So this R side chain could be a lot of different things. In fact, each amino acid has a different side chain. Now, there's only 20 common amino acids in proteins, and we'll look at all 20 of those in a second. So each amino acid has its own name, a 3-letter abbreviation, and a 1-letter abbreviation. So that can make it really easy to list out a chain of amino acids. You can just use the 1-letter abbreviations. So we'll take a look at those as well. All right, now I mentioned that each amino acid has its own unique side chain, which we just kind of write as R if we're writing the generic form of an amino acid. So amino acids can be characterized into 4 groups based on that side chain and its characteristics. So for instance, some amino acids can have a neutral and non-polar side chain. So that would be a hydrocarbon or alkyl group side chain, like a methyl group. If an amino acid has a neutral and polar side chain, then that's another group. So for instance, if you have an OH group, which is polar on your side chain, then that would be another category for that amino acid. You could have a basic side chain, which means that you have, let's see, for instance, a nitrogen that could grab onto another proton. Or you could have an acidic side chain where maybe you have a carboxylic acid that can give up a proton. So the 4 groups are really nonpolar, polar, basic, and acidic. So down below, for example, we do have the amino acid called cysteine. And notice that its 3-letter abbreviation is CYS, so just the first 3 letters of the name. And then its 1-letter abbreviation is a capital C. So that one's pretty easy. Some of the other amino acids have more confusing abbreviations, but we'll get to that. So if we look at the R group here, which is highlighted in blue. We have CH2 and then SH. So sulfur is a fairly polar atom. So this would likely go under category 2, neutral and polar. All right, now let's look at the one next to that, glutamine. So glutamine has the three-letter abbreviation GLN, which I guess kind of makes sense. We've got that GL and then N. But then its one-letter abbreviation is Q, and that's because the G was already taken by another amino acid. But glutamine's side chain does have an amide group at the bottom, so we've got this carbonyl group there. And then we have our NH2 group below that. Now that nitrogen could potentially take on another hydrogen depending on the solution. So it is basic. So remember, bases can grab protons from other molecules. Acids donate protons to other molecules. All right, and then let's look at asparagine. So asparagine has the three-letter abbreviation ASN, which again kind of makes sense. We've got an A, an S, and an N in the name. But then its abbreviation, or one-letter abbreviation, is N. So it doesn't really make sense, but A was already taken by another amino acid. So, all right, so now if we look at this side chain, this is kind of similar to the one we saw for glutamine. There's a nitrogen there in the amide group, and that could act as a base and take another proton. Okay, so let's look at all 20 of the common amino acids. So again, we've got non-polar amino acids, which are in yellow, polar amino acids, which are in blue. Then we have amino acid side chains with a positive charge. So those would be basic, because they took on an extra proton. So let's just write basic here. And then we have amino acid side chains with a negative charge. So that means they're acidic because they gave up a proton to create a negative charge. Okay, and you can kind of tell by the names for the acidic ones. We have aspartic acid and glutamic acid. So it's kind of in the name there, which is nice. Okay, so looking at the 20 common amino acids, you can also see the three-letter and one-letter abbreviations for each. Now, because this quarter is really short, I'm not going to make you memorize these 20 amino acids and their side chains or their codes. But I will want you to potentially identify whether something is nonpolar, polar, or maybe basic or acidic, for instance. All right, so out of those 20 amino acids, 19 of them are chiral. So the alpha carbon in particular is chiral. There is one exception, that is glycine. So glycine is our probably simplest amino acid because the side chain is just another hydrogen. So because this alpha carbon is bonded to two hydrogens, it is not chiral. So remember, a chiral carbon has 4 different groups attached. Now, if we look at the amino acid to the left, that alpha carbon is chiral because it's bonded to 4 different things. Some sort of side chain that's not hydrogen, a hydrogen, a carboxylic acid group, and an amino group. Now, with very few exceptions, amino acids in living systems are in the L form. Now, the way we're going to draw the L form is by having the amino group on the left. So that's pretty easy to remember. Amino acid, L means amino on the left. Now remember, it's the opposite for sugars, right? In living systems, we typically metabolize D sugars, or D monosaccharides. But with amino acids, we use L amino acids to make proteins. Okay, so let's talk about why we have positive and negative charges on amino acids. Amino acids exist as what are called zwitter ions. That's one of my favorite words, is zwitter ion. It's a cool word. So what this means is that you have a molecule with two charges that are opposite from each other, right? So they balance each other out. So you end up with a net charge of 0. So the net charge of a zwitter ion is 0. So if we look down below, on the left, we have the non-ionized form of the amino acid. So that means it's completely neutral, no charges on anything. All atoms are neutral. Now, this doesn't actually exist in our bodies because remember, our bodies do have acidic parts, basic parts, etc. So amino acids actually exist as this zwitter ion form. So the nitrogen, remember, is basic. So it can grab onto a proton and create NH3 plus on the side. And remember, carboxylic acids are acidic, so they can give up their proton and become CO2 minus. So this doesn't necessarily show how it works, but you could think of it as the carboxylic acid is giving its proton to the nitrogen. So they're kind of exchanging that proton. That's not necessarily how it works, but one easy way to remember it. Now, let's talk about what's called the isoelectric point. So this is the pH at which an amino acid has a net charge of 0. So if you have an amino acid with a neutral R group, the isoelectric point will be at about a pH of 6. If you have an acidic R group, so that side chain is acidic, so that would be, for instance, if we go back, that would be either aspartic acid or glutamic acid. Their isoelectric points will be at a pH less than 6. And then if you have a basic R group, so that would be pretty much anything with a bunch of nitrogens. So if we look at like lysine, arginine, histidine, notice that there's these nitrogens in the side chain. So, and they're capable of taking on an extra proton. So then their isoelectric point, where they have a net charge of 0, that would be more than a pH of 6. So this actually makes sense, right? We know that a neutral pH is around 7, but for amino acids, we're going to say it's around 6. Anything less than that, we know from the pH scale, is acidic. So if we think about the full scale from 0 to 14, the middle is 7. Anything less than that is acidic. Anything greater than that is basic. So this makes sense, right? Okay. Now, what's great about this is amino acid solutions can act as buffers because they react with both acid and base. So we've got that amino group on the left that can act as a base, and we've got that carboxylic acid group on the right that can act as an acid. So remember, buffers are solutions that maintain a certain pH by reacting with acid or base depending on what is present in solution. All right, so let's talk about some amino acid properties. So remember, we have amino acids, and they do not naturally exist as a completely neutral molecule. We have typically a positively charged amino group and a negatively charged carboxylic acid group. So they do exist as zwitter ions. So because they're zwitter ions, they're typically found in the form of a solid. So kind of like salts, right? Remember, sodium chloride is a salt, and that exists as a solid, and it has positive and negative charges. So that's kind of similar to an amino acid here. So they're typically white crystalline solids. Because they're solid, they have relatively high melting points. But if you do dissolve amino acids in water, they have a really high solubility because of their positive and negative charges. So let's just kind of draw out a generic amino acid. So we've got our positive charge on the left for our amino group, the negative charge on the right, and then some sort of R group coming off that alpha carbon in the middle. So the positive charge on the nitrogen will be attracted to the partial negative charge on the oxygen for water. And then the negative charge on the carboxylic acid group will be attracted to the hydrogen, the positively or partially positively charged hydrogen on water. So water can easily dissolve amino acids. All right, so let's wrap up this introduction to proteins by kind of going through some questions. So the first question is, how many common amino acids exist in proteins? The second one is, the amino acids in living systems are in the blank form. The next one, the isoelectric point is the pH at which an amino acid has a net charge of blank. And last, in 19 of the 20 amino acids, the alpha carbon is blank. So see if you can complete those sentences and answer the first question, and then we'll go over it together. All right, let's start with the first question. How many common amino acids exist in proteins? 20. That's kind of an easy number to remember. All right, and next, the amino acids in living systems are in the blank form. That should be L. The isoelectric point is the pH at which an amino acid has a net charge of 0. And in 19 of the 20 amino acids, the alpha carbon is chiral. Now, bonus question, which amino acid is not chiral? Glycine. So glycine is the one amino acid that is achiral, which means not chiral. And remember, that's because the alpha carbon's side chain is another hydrogen. Okay, so we'll stop there. Next time we're going to start diving into some amino acid reactions, because that's going to lead us into how proteins are formed. So I will see you then.